Hey guys, today we are going to be covering chapter 3, um, looking at the chemistry of organic molecules. Now, I know we already talked about the um, general chemistry stuff in chapter 2. Organic chemistry is a little bit different in terms of kind of what our focus is on as far as chemistry goes. So when we look at organic chemistry, when we look at organic molecules, we're looking at molecules that actually have carbon and hydrogen both. And because of those carbon and hydrogen bonds, they actually end up forming long hydrocarbon chains. Oftentimes, organic chemistry is sometimes called the chemistry of life science because most of these um, organic molecules can be found in living creatures because of their strong and stable bonds. So the molecules that we care about, the organic molecules that we care about, often are called biomolecules or macromolecules. These are the molecules that we're super familiar with. These are not unfamiliar to us. So, and I know that you've thought about them, at least some of them. So those biomolecules are carbohydrates, lipids, proteins, and nucleic acids. So maybe we don't exactly count nucleic acids per se, but I've definitely counted a carb or two. So um, we will focus in this chapter for the most part on like the different behaviors of carbohydrates, lipids, proteins, and we'll talk about the different structures and also the different shapes as well. Carbon itself is a really unique atom because it's the thing that allows life to have such diversity in a way. Like this molecule has such unique bonding properties. Um, it has 6 electrons. There are 4 electrons in the valence shell, and it needs those 4 atoms or those 4 molecules in that valence shell to be happy. So, in order to uh, have those um, valence shells all full of those 4 electrons and happy-go-lucky, they become quite willing to share those electrons. So because of the willingness of carbon to share with other carbon atoms, they end up making these very strong and very large carbon chains. Um, and we see this quite often in um, other elements as well, other very important organic elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, also known as CHNOPS, not schnapps, the friendly drink, CHNOPS. So carbon can share with other carbon atoms, and it forms those long stable chains called skeletons or backbones. I just talked about that in the last slide. And they can form double or triple bonds even. When we're looking at organic molecules, we're super lucky because we don't have to memorize the shape of them. We don't have to do any mathematical equations. What we really care about is that we know that they have these long carbon change, long carbon technically hydrocarbon chains, I guess. And then they have this functional group. And the functional group is the group of atom combinations that are bonded to those carbon skeletons that allow that molecule to behave in a certain way. Um, and once that functional group is attached to the carbon skeleton, it changes the function of the molecule. So, like a hydroxyl group to a carbon skeleton forms an alcohol, but alone, that's not what you get. Nonpolar molecules can then become polar when this happens. This is just an example of what those functional groups look like. And what you're not seeing is those long carbon chains that are associated with these functional groups. So there are different forms of biomolecules. Essentially, there can be biomolecules, these organic molecules, that are identical in formula but different in arrangement of the atoms. So that can be very confusing. Um, Often organic molecules will join together, um, and you'll see polymers, which are large biomolecules. So that's when you'll see the same unit or monomer linked together. The creation or breaking down of polymers, creation is called synthesis. Um, the breakdown of these large molecules is called degradation, or to degrade it. When you add water, that actually breaks down a biomolecule, so that's called hydrolysis. When you remove water, or it's called dehydration, that actually joins biomolecules together. I can show you an example in lab when we're face to face. Your textbook gives you a cute example. That's not nearly as qualified as my drawings, but it works. And this shows you where hydrolysis splits apart a polymer, and a dehydration synthesis reaction combines a polymer. So when we talk about the biomolecules themselves, there are 4 biomolecules that we focus on in this chapter. So there's the carbohydrates, lipids, proteins, and nucleic acids. The carbohydrates, and we'll talk very briefly about each one of the biomolecules because we got to know a little bit about them. So carbs are, of course, sugars. They're our immediate energy source for life. And so we can see those in various different forms, like monosaccharides, which are simple sugars, with carbon backbones of 3 to 7 atoms. These are kind of a short-term energy source. And there are other examples like glucose, which we find to be delicious apparently. Um, and then there's ribose and deoxyribose, which I'm going to say this now. These are very important to us now, and we'll talk about them again later because these sugars are a component of something very, very important, which is DNA and RNA. So a disaccharide is a 2-unit carbohydrate. So instead of a monosaccharide, mono meaning one unit to make that sugar, disaccharide is 2 monosaccharide units that would be joined together. And so this is a quick energy source, and that includes examples like sucrose, table sugar, lactose, milk sugar, which some people have an issue breaking down milk sugar. And then the polysaccharides, you'll see when you have many monosaccharides joined together. So these are usually acting as long-term energy sources. So like starch in plants, that's a long-term energy source, or glycogen in the liver of animals. So if you look, this is a potato, and then there's these little starch granules. You'll actually get to see that in lab. Cellulose can be found inside plant cell walls, so that's actually a structural polysaccharide. Or chitin in fungus and the exoskeleton of arthropods, also a structural polysaccharide, so something that makes it much stronger. Lipids are another biomolecule that we have to talk about. Even though we don't think fats are important, they really are. Um, they have various important jobs to accomplish even in our bodies alone, which is why we tend to store them a lot. They're water insoluble. Of course. We know that oil and water don't mix. That's what the thing says. And you can see unsaturated fats or saturated fats. For animals, you'll see one of the important fats is triglycerides, and that's really good for long-term energy and insulation. For plants, the uh, plants store their fat as oils instead, and those oils provide long-term energy. A phospholipid is a type of lipid that we see great importance in, especially when we're talking about the plasma membrane of a cell, because the plasma membrane of a cell is a phospholipid bilayer. So if you look, here's your cell, here's just that plasma membrane around the cell, and that shows you how each of these little lipids are joined together, these little phospholipids. And so if you look, um, that phospholipid, there's a polar head, there's fatty acid nonpolar tails. So these tails think water is icky. The head loves water, it's fine. And so the heads will orient towards the watery inside of the cell or the watery outside of the cell. And that leaves the tails that are water hating pointed towards each other on the inside where there's no water. There are other important lipids that we focus on, like steroids, that are kind of used as hormonal messengers in animals, like testosterone, estrogen. And there are waxes, which are used for protection. We like waxes, of course, for candles and things like that, but we really need waxes. If you're a plant, you can prevent water loss. Um, if you're a bee, you want wax to protect your hive. If you're an ear, you need wax in there to kind of protect you and catch all the flying debris that could go in there. I know it's gross, but still clean your ears, but know it has a purpose. Proteins are polymers of amino acids. This is an important biomolecule for a lot of reasons. These guys have a ton of important jobs. In fact, if you were to say, well, what causes that to do that? I would probably say, I bet it's a protein. Like, if you have a really, really serious allergy, usually my thoughts are, I bet it's a protein allergy. And typically it is. Proteins are that valuable. So if you look, I would really note the functions of proteins. So some of those include metabolism. So enzymes that are proteins act to speed up chemical reactions. Of course, there are support proteins like keratin and collagen. There are transport proteins that help move things inside and out to the outside of the cell or vice versa. There are even defense proteins where we have antibodies to bind antigens to protect our cells. There's regulation proteins, which are, of course, hormones. And, of course, proteins aid um, in the function of motion, right? Like actin and myosin allow muscle contraction. So proteins are made up of amino acids, we said that earlier, and there are only 20 different common amino acids. So there's 9 essential that we have to consume, and we have 11 non-essential that we can make. There's only 20 different common amino acids. You can have up to 4 levels of protein structure. So proteins are great when they're like amino acids and they're all linked together in a long chain. However, proteins are not going to work correctly unless they're folded the right way. So you can see a secondary protein you see a polypeptide folded in a 3D shape. Tertiary, where they're more intact. Quaternary, which is like a superstructure, so there's 2 different polypeptide strands. You can see that image in here. It reminds me of a phone cord story. So proteins can become denatured or lose shape if exposed to heat or chemicals. So that can cause a protein to denature. And an organism will die if too many denatured. Think about proteins that have been misfolded, like prions that are misfolded proteins that cause other proteins to fold incorrectly. And that's what's responsible for like mad cow disease and things like that. So nucleic acids are the last biomolecule that we um, talk about. These are polymers of nucleotides. So a nucleotide is a 5-carbon, you'll see a 5-carbon sugar, which we just talked about earlier, a phosphate group, and a nitrogenous base. So the sugar that we talked about earlier was deoxyribose that you'll find in DNA, and ribose that you'll find in RNA. And then there's the phosphate group. So this molecule actually forms a strand. So you'll get a backbone, per se, of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. And then the bases kind of project off the side of that backbone. So you'll see the DNA is a double-stranded molecule, which forms a double helix. RNA is a single-stranded molecule. So polymers of nucleotides form nucleic acids, DNA, and RNA, which we have a whole chapter designated to DNA and RNA. And they can also have the job of carrying energy, like ATP. They can have a job in cell signaling or acting as cofactors, like coenzymes A, FAD, NAD, NADP. Those are going to be very important coenzymes that we talk about later when we talk about photosynthesis and cell resp. So if I were to compare, and there's nothing I love more than a good compare and contrast, if I were to compare DNA to RNA, you could compare, of course the name is different, deoxyribonucleic acid to ribonucleic acid. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. The nitrogenous bases are even different. So DNA actually has adenine, cytosine, thymine, and guanine. And RNA has adenine, cytosine, guanine, and uracil instead of thymine. DNA is responsible for carrying all the genetic information for assembling proteins, and those proteins get expressed and become the people that we are. And RNA just has a big job in, it has many other jobs, but RNA's probably biggest job is protein synthesis and the regulation of gene expression. This shows you the example of those nitrogenous bases that stick off the side of that sugar phosphate backbone. So you'll see pyrimidines and purines. So the pyrimidines are cytosine and thymine. They're just a little bit bigger and different shaped than the purines, like adenine and guanine. So DNA is pretty critical and forms this double helix thing, but the real reason why it's available to form this double helix is because of complementary base pairing. And when one strand of DNA makes a complementary base pair with another strand of DNA, they'll join together in a hydrogen bond, okay? So when you're looking at complementary base pairing, adenine will always pair with a thymine or uracil if it's RNA, and cytosine will always pair with guanine. There's no more reliable relationship than cytosine to guanine, guanine to cytosine. It's consistent across the board. So there's just a little bit more information about DNA. I do mention that we have a whole entire chapter designated to DNA later that we'll focus on. Their RNA has a big starring role also in the chapter whenever we cover transcription and translation, or protein synthesis. ATP is another important nucleic acid. It's adenosine triphosphate. It's called the energy currency of the cell because we're able to take ATP and break it down and derive energy from it.